Well, thank you, Dr. Strauss, for that wonderful introduction. I would add to what you say that there are many ways to suffer, and we all engage in all of them. So anyway, it's my pleasure to come here and to give you an update on the Tafazzin enzyme replacement therapy that I've been working on. Let me thank the organizers for inviting me, certainly thank the foundation for funding the early part of this work. And thank you all for coming, patients, families, and scientists, because it's always hard to speak in the middle of the afternoon after lunch, but before the coffee. So I'll try to keep you entertained, and if you need to go get coffee early, it's okay. Anyway, before I launch into the science, let me just disclose that I am the founder of a company called Transcellular Therapeutics, and so everything I say has to be colored in that context. But the company was founded just to try and commercialize this therapy so we can get it to patients faster. Anyway, many of you know the background, so I've actually changed my talk somewhat in that I'm not going to give a background about all the biochemical abnormalities, et cetera, because many speakers have come and gone and done a fabulous job of introducing it. I thought I'd first start by sharing my perspectives on developing a treatment for Barth syndrome. And again, to compliment all of you for supporting this work, because 3 years ago when I started this, it was just a dream. I had no data, and I wrote a grant, and I got it. And, it's been great since. When I started this project, it's a funny story, which I guess I should tell you guys at some point about how I got into it. I'd never heard of Barth syndrome. But I actually saw your newsletter, and it's not because I got the newsletter, it's because my wife got the newsletter, because she studies neutropenia. And she showed it to me, and I said, oh, this is fascinating. They have a foundation, they have a grant mechanism, I'll apply, great. Anyway, and the rest is how I got into it. Anyway, at the time where I wrote the grant, I realized there was really no efficacious therapy. Let's see, I think I have to favor people on this side of the room, because everyone's been going to this side, so I'll show on this side. There really was no efficacious therapy available at the time. I think it's exciting because we're hearing about all the possibilities that are about to come. Care still is really mostly supportive. Standard heart failure therapy, ICD placements, heart transplants. And really there's been no enzyme replacement therapy available. And so I viewed that as an unmet need. And I found out that there potential therapies can be tested, as many people are doing, in a commercially available mouse model, this doxycycline-inducible knockdown of Tafazzin. Oops. And the idea I had was very simple, and that's just to take Tafazzin, the enzyme, and to manufacture it in E. coli through recombinant means, and then tether it to something called a cell-penetrating peptide. Okay, equal time for this side. Recombinant Tafazzin by itself is made inside the cell and has to localize to the mitochondria in order to do its work. And so how could we give it as a drug? Well, the answer is you really couldn't. If you gave it to someone intravenously, well, it wouldn't go into the cells because it has no mechanism for doing so. However, if we tether it to something called a cell-penetrating peptide, that peptide theoretically would confer upon it the properties where it could be administered systemically, it could cross the cell membrane, enter the cell, localize the mitochondria, and if enzymatically active, rescue the cardiolipin remodeling defect and rescue the mitochondrial respiration. There's a large body of literature on cell-penetrating peptides. They've been around for more than 20 years, and there are quite many of them. What are they? Again, forgive me, I'll switch to this side again. They're small peptides, generally less than 30 amino acids, often cationic. They confer the ability to enter cells. They're not really cell-specific in most cases. I mean, there are goals to develop those that are cell-specific, and they really don't damage the membrane. They're very versatile delivery vehicles in that they've been used in multiple contexts to deliver nucleic acids, small molecules, RNAs, other proteins, peptides, etc. And they generally share the physicochemical property of being amphipathic. They come from multiple places. Some of them occur naturally, like a sequence in HIV TAT protein or Drosophila antennapedi protein. Some of them are chimeric and others are synthetic. But anyway, there's a large body of literature. Anybody who wants to know more about them can go to this website called CPP Site 2.0, where they curate all the known cell-penetrating peptides. Right now, there are about 1,800 or more. When we started this project, we wanted to make sure that from a biochemical standpoint, Tafazzin would be amenable. As you heard from our lunchtime speaker, Tafazzin is a small protein, so that enables us to do this in something simple like E. coli. We all know what it does. It it catalyzes the remodeling of monolysyl cardiolipin to other forms of remodeled cardiolipin. The protein structure hasn't been solved, but it's thought to be reasonably simple just by structure predictions that there's a transmembrane domain and a phosphate-acyl transferase domain. The crystal structure hasn't been elucidated, and the structure-function relationships aren't understood. But an isoelectric point prediction predicts that it has a high isoelectric point, which is important from a biochemical standpoint because it means we can use this property to purify it. As many of you know, there are multiple outcome measures in Barth syndrome to test efficacy, both in mice and in patients. Number one is the MLCL to L4CL ratio. We've seen a lot about mitochondrial oxygen consumption rates. We've seen a number of studies on reactive oxygen species generation, mitochondrial morphology, of course, muscle performance. And then from Johns Hopkins, we've heard about some novel biomarkers, such as plasma 3-methylgluconic acid and plasma arginine. And those are very important as we measure surrogate outcomes as well as clinical outcomes. The other reason for going into this area of enzyme replacement therapy is that it's a path well trodden. When we try to bring therapies to patients, there's something called the translational valley of death. As we try to go from mice to people, a lot of studies fail because mice are different and there are many hurdles that must be vaulted over to get FDA approval. And so for enzyme replacement therapy in principle, there are actually multiple enzyme replacement therapies that are commercially available for various other genetic disorders. And the FDA has actually published some regulatory guidance documents as to what their standards are for FDA approval, so we could use those documents as we design clinical studies. Anyway, so our experimental approach has been conceptually very simple. It's to design these recombinant Tafazzin proteins with cell-penetrating peptides, synthesize and purify them, which sounds easy, but it's been more challenging than it sounds. Demonstrate uptake into the cells, demonstrate the enzymatic activity inside the cells, such as cardiolipin modification and mitochondrial respiration, then ultimately rescue the cardiomyopathy and the skeletal myopathy in the mouse model. So, our design was to take the mouse full-length protein, which is 262 amino acids, and make 3 versions, an untagged version with no cell-penetrating peptide, and then conjugation to either antenna PDI or something called CTP, which stands for cardiac targeting peptide. That's a synthetic cell-penetrating peptide that was identified in a phage display screen, not by my lab, but by another lab. We also made the human version, thinking that, well, this might be clinically useful, specifically the abundant isoform lacking exon 5, again, untagged or modified to contain either antenna PDI or CTP. And we expressed them in E. coli using modified PET vectors, which adds a polyhistidine tag useful in purification and a TEV protease cleavage site so we can cleave off the tag. And so this is just one of our early gels showing how we could actually induce expression. So this is what we call, for the non-scientists, so this is a protein lysate from E. coli, and this is what the basal protein expression looks like, and then this is what the inducible expression looks like when we induce expression with IPTG. And we can see we get a pretty good band. We can purify it with metal chromatography, taking advantage of this polyhistidine tag, and we could really purify it in milligram quantities. The big problem is it's very insoluble, so we can make it in milligram quantities, but it's in something called inclusion bodies, which is just a big glom of protein that is very hard to work with. So what we've been able to do, and again, to give credit to the people in my lab, is because it took us months, is that we could dissolve them in denaturants. But the real key was developing a renaturation protocol where we could renature these proteins and keep them soluble and active, and that took us a long time. But the good news is we've been able to do it for multiple versions of these proteins, from both human and mouse. We have Tafazzin without the tag. We have 2 versions of human Tafazzin with CTP and with antenna PDI, and we have a mutant version of Tafazzin with CTP, and same with mouse. We have all of them, and we've been able to make them and renature them and test them in cells. And one of the first tests was to say, to ask the question, does this Tafazzin localize to mitochondria? So this is just an immunofluorescent stain looking for protein and also its presence in mitochondria. So the green signal actually is looking at It's an antibody against Tafazzin, commercially available one from Sigma. This is Tafazzin without a tag. This is Tafazzin with the CTP tag. And the mitotracker in red just gives the mitochondrial signal. And you can see with no cell-penetrating peptide, we really don't see any green inside the cells or any overlap between green and red, which should be yellow-orange. Whereas if we look with the CTP version, we can see that there's lots of yellow signal, suggesting that it's going into the cell and localizing to the mitochondria. We've been able to do it with multiple versions of the protein and in different cells. I'm sorry, this was actually H9C2 myoblasts, and these are C2C12 skeletal myoblasts. But you can see here that we also see uptake inside the cell and localization to the mitochondria. We did some pulse chase experiments trying to decide how much we had to give and how long it stays inside the cell. And so what we're looking at here are bacterial expressed Tafazzin and looking at the kinetics of uptake and persistence in H9C2 myoblasts. So this is where we treated the cells with a small amount for 2 hours and then measured and did what we call a chase experiment, where we took the protein away and then waited and tried to see how long it would take before we could see the protein inside the cells and how long does it last. And this is a chase at 2 hours. Again, we used a small amount, so we can see the green is persistence of the protein on the surface, or perhaps presence in lysosomes because some of it gets degraded. The way these peptides work is that the cells are usually taken up by, the protein is usually taken up by endocytosis, and the protein has to undergo what we call lysosomal escape, where it escapes degradation. So it has to exit the lysosome and then travel to the mitochondria, which is challenging to do. But you can see that we do see yellow dots as early as 2 hours after the uptake. And if we look longer, we can actually see, this is just vehicle-treated cells, but if we have the Tafazzin CTP compound, we can see that we can detect these yellow dots out to beyond 18 hours. And then even at 24 and 36 hours, we can still see yellow dots, which is exciting to us because it means when we give it, it actually goes in and it actually can stay there for a reasonable amount of time. And what I also note is that our antibody is pretty insensitive in that it doesn't detect the endogenous Tafazzin, because these are wild-type cells. They all have Tafazzin, but our antibody doesn't detect the endogenous Tafazzin because it's relatively insensitive. So it probably means we're getting a fair amount of the protein into the cells, which is why we can detect it. Hard to be quantitative, though. We're doing that with subcellular localization experiments, which we've done. I actually have some of the data, but I haven't put it in this talk. And we've also developed a LCMS assay that I'll show later. That'll give us even better quantitation. Okay, oh, that's just another picture showing that this is a 24-hour pulse, so just slightly different experiment where we pulse longer and did a longer chase. And again, we see yellow dots after 24 hours, indicating that it stays in the mitochondria for a while. But the real question is, when it goes into the cell, is it enzymatically active? What does it do? So, as many of you have heard today, one of the hallmarks is of Barth syndrome is an elevation in the MLCL to L4-CL ratio. So we've been actually able to isolate lipids from Tafazzin-deficient cells. In this case, it's cardiac fibroblasts isolated from the Tafazzin-deficient mice. And we've been able to measure the MLCL to L4-CL ratio by LCMS, and then we use single ion monitoring to actually quantify the amount of MLCL and L4-CL and then look at the ratio. And you can see that in vehicle-treated cells, we have in knockdown cells, we have an elevation of the MLCL to L4-CL ratio, whereas in wild-type cells, this ratio is much lower. When we treat with Tafazzin that has no cell-penetrating peptide, we do not affect this MLCL to L4-CL ratio in the knockouts or in the wild-type cells. However, when we add the CTP, we do see a reduction in the MLCL to L4-CL ratio compared to the wild-type cells, and same with the human Tafazzin antenna-PDI construct. Now, what about in, well, actually, the next one is, what about oxygen consumption? So this is a seahorse assay, which many of you have seen and are experts at. And so these are also the Tafazzin-deficient cardiac fibroblasts on doxycycline using glucose as a substrate. And you can see from the light green, this is the knockdown cells. And we see lower basal respiration and then lower maximal respiration. When we add our Tafazzin I have to see what, sorry, it's very small. When we add our Tafazzin and PDI or CTP, we actually see augmentation of maximal respiration to above the wild type levels. That's very exciting to us. So it seems to be restoring both the rescuing both the cardiolipin remodeling defect as well as the oxygen consumption defect, at least in cultured cells. The real question is, what does it do in mice? And that's actually a trickier question. And so what we did is we did this crazy experiment where we just took these Tafazzin deficient mice and we treated them very rapidly for 8 days because, you know, that's what my graduate student decided to do. I have no control over my graduate students sometimes. And so what she did is she treated the mice for 8 days continuously with just a high dose of this recombinant Tafazzin. And lo and behold, she was able to show that these mice, after only 8 days of treatment, had a reduction in the MLCL to L4CL ratio. And we've since done it a few more times. And so what you can see is that in vehicle-treated mice, again, as expected, they have the knockdowns have a very elevated MLCL to L4CL ratio, whereas the wild types have a low MLCL to L4CL ratio. Mice that are treated with a CTP compound, there seems to be a significant decrease in the MLCL component. And then, you know, we haven't done as many with N-tended PDI, but it does look like, well, in one of 2 mice, it actually is lower. Again, we have to do this more, but it's still very exciting that we can actually see a change with just such a short treatment. I think this doesn't necessarily model what we would do in patients or in the disease, because realistically what we want to do is chronic treatment and see if we can get a long-lasting benefit. Anyway, we also looked at the mitochondrial morphology in the hearts from these mice that were treated. And, you know, this is just a lower power view. And, you know, it's hard to see, but we see what other people have reported with these mice, that there seems to be like a dense stacking of the intermitochondrial membrane in the knockdown cells. And perhaps after treatment, the density is a little bit lower. And I think this is a little bit more apparent in high power. Again, it's hard to quantify this, but this is just something we see. So again, it makes us excited that with just a short time of treatment, we see improvement in mitochondrial morphology. Now, what about oxygen consumption rate? And I show this graph just to give my graduate student credit for doing a lot of studies. This is actually, this is actually oxygraph data that Adam Chico can appreciate, that we've actually looked at oxygen consumption in males and females, wild type and knockdown mice. And as you've been hearing from many people this morning, that these mice actually have a fairly mild phenotype. And in fact, in males, we can't really see any difference in oxygen consumption rate. But we can see a difference, for females. And again, you know, people in the audience can comment, but again, I'm showing you the raw data that this is what we see. And so the next slide just shows more of the female data. And female data seems to be more consistent, that when we compare the wild type versus the knockdown, we see a general reduction in oxygen consumption rate for complex 2. And you know, the difference is biggest for complex 2. The slight difference for complex 1, but it doesn't achieve statistical significance. What was really interesting, though, is when we actually treat, we find that our Tafazzin CTP and our mutant Tafazzin CTP seem to augment the activity of complex 1. We're still not sure what that means, but we're very excited, particularly, but also puzzled, because why would this mutant seem to augment so well? Because the mutant is supposed to be a loss of function mutant. So we actually went back and tried to quantify this mutant in some of our in vitro assays. You know, originally we did it as a control. And what we found is that this mutant actually has significant activity, because this is just going back to look at the MLCL to L4CL ratio in our cardiac fibroblasts again. And if you look in wild-type cells treated with, well, I should say that each set is the treatment, and what we're doing is we're treating homozygous knockdown, heterozygous knockdown, and then wild-type cells. The wild-type cells each have the lowest MLCL to L4CL ratio, whereas the vehicle-treated each has the highest MLCL to L4CL ratio. And you can see that compared to vehicle, all the treatments seem to reduce the MLCL to L4CL ratio, but our mutant Tafazzin seems to reduce it the most, which is totally unexpected because it should be loss of function. And so we actually looked in the seahorse assay too, and again, recapitulating what I showed earlier, that both the antenna PDI and the CTP version seem to augment respiration to levels above wild type, this Tafazzin mutant actually seemed to be comparable, if not even slightly better. So, you know, again, we were puzzled, but we're intrigued too, because it means that this mutant may actually function better than expected, because it may convey some advantage in pharmacology. And that leads me to the next stage. So in order to use this as a pharmaceutical, we really have to know how much we're giving and where it goes, and that means that we have to understand this pharmacokinetics, because we'll never get FDA approval for this as a drug without understanding this. So we had to develop what we call a pharmacokinetic assay, where we could track what happens to it, where it goes, how long it lasts, how much is taken up in the organs. And so we did this using an LCMS proteomic assay, where we can detect unique human Tafazzin peptides. And so we did the experiment where we detected the peptides in mouse plasma first, and the idea is we're going to do it in mouse tissue as well. And so, the studies are ongoing, but we're very excited. So in looking at the peptide and doing some preliminary studies, we were able to identify one particular peptide that we can detect with high sensitivity that actually is unique to human Tafazzin and different from mouse Tafazzin. And we were able to do some spiking experiments where we spiked mouse plasma with varying amounts and developed these standard curves. And you can see that this peptide actually is distinguished from the mice. It's not distinguished from non-human primates, but we have another one that distinguishes it in non-human primates. And that's important for some primate pharmacokinetic and toxicology studies that we have on the drawing board. But what was nice about this assay is that we can detect it in the attomolar range, which means it's very sensitive. And so we've actually done some preliminary experiments in mouse plasma where we've actually infused it into mice, and we can detect it in the blood, 1 hour after we inject it at still femtomolar concentrations, which means that it's detectable, but it's probably being rapidly cleared. Where it's going, we don't know yet, so we're still trying to go through our organs and our whole blood to see if it's in the leukocytes or where it actually goes to. So that'll probably be a topic for the next time I talk to you. Anyway, the other caveat with enzyme replacement therapy or the possibility of immune responses, because basically we're injecting a foreign protein. Luckily, Tafazzin is highly conserved. There aren't much differences, so hopefully there's not much immune response. But again, we just don't know. So what we actually did is some preliminary experiments looking for an immune reaction in mice. So what we did is we took a cohort of mice, we collected pre-immune serum, reprimed the mouse with mouse Tafazzin linked to different CPPs. We boosted with the same protein after 6 weeks, and then after the boost, we collected serum 2 weeks. And then we tested the serum for antibodies against a recombinant protein panel using an ELISA assay. And so this is just a very busy slide, and it's probably too small for me to see from here, so I'll have to look at this screen. What we basically have on the bottom is we have different mice grouped by number, and then also indicating what they're injected with, and then whether it's pre-immune or post-boosting. And our first green line here is just mouse plasma spike with a commercially available monoclonal antibody against Tafazzin as a positive control. And I think what you can see is that, well, certainly all of the pre-immune or vehicle-treated mice really don't develop much of an antibody response. If we look at the various CTP mice, number 1 really didn't develop much antibody response. Number 2 didn't develop much antibody response. Number 3 was a little funny. So if you look at our panel, we have, let's see, is that showing? I don't even know if that's showing. Yeah, we're missing some of the labels, so I apologize. We actually have a panel of, how many proteins? Like 7 different proteins that don't overlap in their epitopes. And so the third mouse is reacting kind of strangely because it's reacting against proteins that have no overlap in protein sequence. So we actually think that this group is probably reacting to some sort of contaminant because these proteins don't overlap in sequence. In fact, I think the blue one doesn't even have Tafazzin in it. I think it's actually a Cree protein. And the fourth mouse really didn't develop much response. The antenna PDI mice are a different story. 2 out of 3 mice actually developed significant antibodies against Tafazzin. So the cell-penetrating peptide really seems to make a difference as to what kind of immune response we develop. All of you are going to ask how there's so much variability in this mouse model, and that has indeed been our experience. You know, we've aged these mice for a long time. I think most recently we went to 17 and a half months before we cried uncle. But they really don't develop a decrease in ejection fraction or fractional shortening. Again, we are using anesthesia, but we make sure that the heart rate is above 500. And again, the fractional shortening is usually doesn't go below 40% either. But we just don't see any heart failure that develops over time. We did try to induce heart failure using a transverse aortic constriction model where we put a suture around the aorta to induce pressure overload. We didn't see any heart failure, but 75% of the mice died within the first couple of days. So, you know, maybe we need to fine-tune our model, but I think that this is a challenge for all of us in the field. So anyway, let me summarize, and I think I will get you to coffee soon. We made these recombinant tafazzins engineered for cellular uptake, and they've been synthesized and purified. They enter the wild type and tafazzin-deficient cells, localized to the mitochondria. They improve the MLCL-to-L4-CL ratio, and they rescue the defective respiration. We can deliver them systemically to the tafazzin-deficient adult mice and see improvement in the MLCL-to-L4-CL ratio. And we think they're improving respiration, but we really haven't optimized the pharmacokinetics. So I'm hoping that I'll have more effectiveness in the future. We have a lot of future work to do. I think we need to choose our lead candidate based on the activity in vitro, in vivo, and perhaps the immunogenicity. We need to optimize the dosing in vitro based on the pharmacokinetic studies. You know, we need to rescue cardiomyopathy in this mouse model, so we have to figure out how better to induce it. Because we have so much recombinant protein, we've been talking with an X-ray crystallographer about determining the crystal structure and also determining the structure-function relationship. I was fortunate enough that I got a grant to do a small pilot study in non-human primates for pharmacokinetics, immunogenicity, and toxicology, so I'll hopefully be able to tell you about the next Barth syndrome meeting. And then we're also looking to determine the ability to rescue heart failure in other non-Barth syndrome models because, as many of you know from Dr. Spiragno's work, tafazzin seems to be reduced in other forms of heart failure. And that is one of the selling points of developing this commercially because Barth is a small market, but when I tell the various entities, I say, well, but it also seems to be reduced in other forms of heart failure, so you may get the orphan indication, but you may be able to have a secondary indication later. So that usually changes the conversation for the better. Anyway, still ongoing though. And obviously I've been talking to a lot of people here about developing a pathway to clinical trials because that's really the bottom line. We'd love to get it to phase 1 within 2 years. Anyway, obviously I didn't do this work myself. I think that much of it has been done by my graduate student, Anna, and I'm not going to let her graduate till we finish all this stuff. And my research scientist, Wei-Ming Chen, as well as my surgeon and echocardiographer, Yonggang Liu. Collaborators, David Marsnick, who knows far more about mitochondria than I do and lets us use his oxygraph as long as we don't break it. Dale Whittington in the mass spectrometry core does our LCMS for cardiolipin. Phil Gafkin in the FHR, in the Hutchinson Cancer Research Center proteomics core has helped us work out the peptide assay for pharmacokinetics. And of course, the Barth Syndrome Foundation. Without you guys, this would not be possible. Anyway, I'll stop here.
Thank you, Michael. A couple of questions.
Thank you for the talk. Very elegant approach to enzyme replacement, and your lipid and bioenergetic data certainly seem hopeful and compelling. My question is, how on earth is this recombinant protein actually getting to the right submitochondrial localization? Because attaching it to a cell-penetrating peptide seems to be a pretty way to get these things across the plasma membrane. But a couple of points on getting it into the intermembrane space, right? So first of all, you need if there's a targeting peptide to go through the TOM complex, by all indications, these things have to be completely unfolded to get through this roughly 20 angstrom diameter transmembrane pore. So that's one. Second of all, if it's getting taken up by endocytosis, there's really not much indication that mitochondria are part of the endomembrane system. So how do you think it's going from an endosome to the mitochondria? This seems to be a leap of faith to get this into the right mitochondrial localization.
Those are great questions, and you're absolutely right. It's a leap of faith because I think for a lot of the cell-penetrating peptide data, basically they work somehow, and people still are arguing over the mechanisms. And they postulated things like endocytotic mechanisms using various endocytotic pathways, but also inducible pore mechanisms. And so we don't really know. And you're asking me to speculate, but I can't really speculate because we don't have any data. And so theoretically, you know, there's the school of thought that most of it happens by endocytosis. They go to lysosomes. Somehow they escape from the lysosomes, but then, as you point out, how then do you get to the mitochondria? And the answer is, I really don't know. But I will say there's hope in that, you know, I wrote an R01 to ask exactly that question, what happened, what's the intracellular trafficking? And, you know, hopefully I'll be able to tell you that soon. So, but those are great questions, and I can't say I can answer them.
In the mass spectra that you showed us, or in this one mass spectrum, L4 cardiolipin is a very minor component in your heart. And that's consistent with what our people have found in mouse heart. So why do you choose L4 cardiolipin as your denominator? Because it would be rather make much more sense to choose a major cardiolipin species.
Yeah,
and also related to that, my question is, did you see any changes in the cardiolipin molecular species pattern?
Right. So that's a wonderful question as well. So we, in our single ion monitoring, we actually look at 4 species. We looked at 747, we looked at 723, we looked at 594 and 582. And so, you know, when we look at the total, you know, we looked at total modified cardiolipin and total monolysocardiolipin. You know, at least in these early experiments, it doesn't seem to change that much when we look at total. But again, I feel that it's not, whereas we do see the biggest change in the L4CL and the MLCL. And so that I have to say that I think there's a caveat here that we haven't really optimized the assay, the pharmacokinetics. So I think that this is just to say that we should keep working, but I think your points are all well taken.
Okay, thank you very much.